Okay, hi guys. Wanted to talk a little bit about genomics. So kind of wrapped up the basics of the nucleic acids and do look into viruses. What are they made of? There's DNA core, got a little picture here, and wrapped with a protein coat. Let's see here. Share this with the camera. So there's a picture in your book, but look up on Wikipedia, wiki viruses. And basically it's a squiggle of an internal core of either DNA or RNA. And then it's got a protein coat or a capsid. And that's mostly it. Some of these actually have like fancy landing gear and injection device. And then coronaviruses, they've got these spikes on them to help them dock, and they have actually a lipid coat, lipid membrane coat. So the main thing is they've got nucleic acids, DNA or RNA, and the protein coat. Those two you must remember. So nucleic acids, DNA or RNA on the inside, and they're not just naked, exposed to the world. They're wrapped in a tough little protein coat, okay? Those are two essential ingredients. So viruses can't copy themselves. They have to go into cells to do it, as was shown in our book last time. The protein capsids not only protect them, but sometimes they have little attachment feet and actual injection device. In particular, viruses, well, they can't reproduce themselves because they're not alive, really, they're not cells. So they have to hijack cells to do it. But viruses don't just attack human cells, they attack all kinds of cells, and especially when they attack bacteria. So look up bacteriophage. Bacteriophage means like to eat. So these are viruses that essentially attack bacteria. And in particular, maybe look at the T4 phage, the T4 bacteriophage. It's remarkable. The picture of that, it's incredible. We'll have a peek later. All right. So antibiotics do not work against these because they're not alive. But we can use antivirals. So a lot of times antivirals interrupt nucleic acid synthesis. Where's the nucleic acid? DNA and RNA. The NA stands for nucleic acid. So antivirals often attack that or the enzymes used to rebuild the virus. Okay, that's kind of review. So we're going to launch into genomics today, where we have a book that has an actual chapter on that. And I did revise, yet again, a simplified study guide. So I replaced that on Alima. Please have a look at that. So I redrew this diagram I want us to know, but you have the one that we drew before from the previous chat. And I expect you've still got a copy of that. Have a look at that later. And so this is a review of that. And we're talking about going from DNA, copies itself, replication, transcription to RNA, translation to protein, post-translational modifications to functional proteins. And these are some examples of functional proteins. Hemoglobin has iron in it, a metalloprotein in it, carries oxygen, enzymes, all kinds of things. Many have metals or water-soluble vitamins, receptors, like that. So we're gonna we've gone through the nucleic acids. Now we're gonna talk a bit about genomics. It's sort of a bigger view of DNA. And we'll have a little discussion on GMOs, genetically modified organisms. So I've reordered these things and I've put body fluids next. And if we have time for it, we'll get to molecular messengers. My favorite topic and what my doctoral degree mostly is in pharmacology, well, and toxicology, but we have to do body fluids. And remarkably for pre-medical books, there are only a few that have a body fluids chapter, and it's really useful for that. And if you're on the farm and you're trying to take care of your plants or animals, they've got body fluids too. And you can tell a lot about their health status by checking their body fluids. Okay, so there's a reordering of that. And molecular messengers will leave for the last, if we have time. Alrighty. Okay, so what is genomics? The genome, for starters, I hope you've been doing your homework, and please do notice I put some new videos online for you guys on our YouTube playlist. So the genome, what is that? Here's a little picture of some DNA in a test tube. I worked at a company that made custom lengths of DNA. We jokingly call them designer genes. So DNA, as you know, it's a really long molecule, and it's written from one direction to another, 5' to 3'. And last time we talked about the introns and exons. So if we want in English to say Mary had a little lamb, we see that the words are very nicely separated. We read left to right, so we know how to interpret that. But look at the line below that. This is how it actually, that same phrase would occur in DNA. It looks like a bunch of gobbledygook, and then we see the letters in red, and if we tip them out and splice them together, just like splicing out the introns and concatenating or hooking up the exons, we recreate the Mary had a little lamb saying. So don't forget about that. We have all of those introns. We have to cut them out or excise, and then hook together the exons, concatenate them, and then we've got the message there, codes for the protein with codons. Okay, and remember what percent roughly were exons out of the whole genome? It's a stunner, it's only about 2%. That's just amazing. So again, what is the basic definition of the genome? I wrote it here in huge letters. So essentially, all of the DNA in an organism, the whole ball of wax, right? And I want to talk today especially about genetic markers. So essentially it's a recognizable sequence of DNA. And you can look on Wikipedia, and we'll take a look at that and look in the book. And in particular, I want to spend some time on this diagram of markers of disease. We'll come back to that. But basically, I'm going to go over a couple of basic ideas about the markers. So to start out with, I'll just mask some of this here so you don't have to look at all of it at once. All right, so genetic markers, recognizable sequence of DNA, they can be active or have a biological effect. Some are not active. We don't know if they have an effect, we don't know what it is. But we can trace these markers, and sometimes that's used to trace family history, genealogy, or paternity. So they don't have to be active or expressed as proteins. Sometimes they're just there, and it's a pattern. They talk about a DNA fingerprint. So that's a subset of markers. So we can jot down DNA fingerprinting. And the fingerprint, it's the idea, it's unique. Okay, so we can identify individuals. Right, so the markers they can be active and express proteins or regulate the expression of proteins, some introns do, or they can be inactive. Well, sometimes that just means we don't know the effect. They may have one, we just don't know it. They can indicate a specific biological effect. Now, sometimes that effect might be good or desired, and sometimes it might not. So they can indicate a specific effect, or they can indicate just an increased chance of that effect. So they used to think that if you had the marker, you were going to get the effect. We know better now. It's not that simple. So here's this other little list I'm adding there about genetic markers and their effects. So some effects can be benign, just meaning not harmful. Hair color is an example. It's controlled by a few genes, and there are a couple of types of pigment, melanin, the same that's in skin pigment, and pheomelanin, the brilliant red color, like found in northern Italy and Ireland and stuff. Eye color is also controlled by genetics, but it's controlled by more than one gene. A lot of traits we think of there's more involved in regulating than just a simple gene or a marker, something more complicated. So again, some genetic markers can be markers of inherited disease. So, and again, in some cases, it's if you have the marker, you're definitely going to get that effect. And in other cases, it just means you have an increased chance. So the marker, they used to think if you had the marker, you'd get the effect. And in some cases, that's true. It's a 100% indicator that that person will get that disease. In many cases, it's just, we know that there's a greater than zero chance, which is true for all of us. But having the marker just shows an increased chance. So be very careful about this and discuss it with your healthcare provider, because things that used to be believed even 5 or 10 years ago, well, we have a different understanding. As time goes by, humans change their understanding of the world. That's the whole point of science. For most of human history, people were pretty sure that the world was flat. And now we know that it's not. Okay. So another thing to look at is genome in Wikipedia. I just found out how to hook my iPad directly up to my laptop, but I don't have enough ports to do it and use the overhead camera and the mouse as well. So look these up on your own, Wikipedia, and it's a really good resource. You don't have to dive into the deepest reaches of there, but just, you know, have a peek. Genome size, that's a really interesting thing. How big is the genome? So people think we're really hot stuff and we should have the biggest, most complicated genomes around. But I've got some surprises in store for you. And the ag folks are going to love this. Some plants are more complicated than we are genetically. Okay, here's a picture of what level of DNA structure. These are chromosomes. So we're talking about that as the quaternary level, or the 4th level of structure. Right? Humans have 23 pairs. So that's 46 total. Diploid, that's you got the pair, the set from your mom and your dad. And a haploid is just 1/2, like the set you got from one parent or the other. Okay, so that's Wiki goes diving in deep, and oh my heavens, look how detailed. There's part of a DNA sequence of a virus. Okay, a couple more terms I wanted to talk about. There's, well, you can also look up genetic disorders, which we'll get back to in the book. The book has markers of that, and there are a variety of genetic disorders. And sometimes the genes are very strong indicators, and sometimes they just suggest an increased chance. But that's a deep world, and it's growing and changing constantly. So not all healthcare providers are able to keep up with current literature. Science moves really fast, and healthcare providers in the clinic, their first responsibility is keeping the patient healthy. So they don't always have time to keep up on the science. And sometimes physicians forget that there are many, many more scientists working around the world than there are physicians. And it's usually the scientists who find out the actual way things work. Well, there's a lot of crossover, and some physicians make really good scientists, but they're not really trained. Trained in medicine is not training to do science. Some physicians were trained in science first and can do really wonderful science, can and have. Okay, so here's a picture in our book, and it was talking about genetic polymorphisms. So that was a word I wanted us to know. Genetic polymorphisms, we can expect to see that on the test there. And so what's that mean? You know enough Latin and Greek now to have a pretty good guess for that. Poly is, of course, more than one. Morph means form, more than one form. So our classic example, and we looked at this already, I've drawn this in class back when we were in class. This is the age going along here on the horizontal from birth, say to 1, 2, 3 years old, and then let's have a little gap here, and then maybe up into adulthood, let's say 10 years old, 20 and 30 and so forth. Okay, so this on the vertical would be lactase. What's that? Lactase. What do the last three letters tell us? ASE. From that, we know as trained biochemists, that's an enzyme. And you probably have guessed correctly that that enzyme breaks down lactose. So lactase activity measured in individuals at birth, it's pretty high in everybody. That's really useful because at birth, an infant's main source of food is mom, breast milk, and that has milk sugar in it. So being able to digest that, pretty handy for everyone. But some folks, after they're out of infancy, they don't keep producing lactase because it's not really needed so much. And their lactase activity dies off after, 2, 3, 4 years old. And some argue this is the more evolved races. Asians have often trouble with lactose intolerance. Supposedly humans had tails at one time, or predecessors rather, and we still have a tail bone and the third molar. So Asians tend not to have the third molar. I'm a primitive hairy Frenchman. I've got, a tailbone, well, we all do, and hairy skin, third molar. So other folks, lactase persists longer. That's not necessarily simply a case of more or less evolved, it's differently evolved. And, so there are different races, so to speak, around the world, and their lactase persists. And then there are these folks here, and their lactase stays high pretty much throughout life. And those are, that's found commonly in the Scandinavian folks. Scandinavians, so these are folks that live way up in the north, Norway, Sweden, Denmark, Iceland, like that. And I guess back in the day, if they couldn't survive the winter on reindeer milk, they couldn't survive, didn't live to reproductive age, and who knows? That may or may not be the story, but lactose intolerance is very rare in that group, relatively speaking. So the point here, this is one example of genetic polymorphisms. So that's something we already covered already, genetic polymorphisms. All right, so that's one example. And the chart here in the book, that's their whole name for this whole chart. But I'm preferring that you think of it in terms of genetic markers. In this case, rather than markers of hair color or something, these are markers of disease. So we have markers for hemophilia, for muscular dystrophy. There's malignant melanoma. I probably have that one since I have actually had melanoma. And there's sickle cell anemia. We talked about that. And PKU, phenylketonuria, an inability to process phenylalanine properly. So PKU babies are tested for that. And if they have this enzymatic deficiency, they don't make an enzyme that is useful in metabolizing phenylalanine, they can just seek to have a low phenylalanine diet. But now we can actually, there are gene therapies where it's possible to go in and give them the gene for that enzyme. So here's one huge caveat with this, a couple of them actually. So they used to believe if you had the marker, you would get the disease, full stop, end of story. We now know that's only true for some particular things, not for everything. And as time goes by, scientific opinion is evolving. So check with your healthcare provider, look into Wikipedia, do some research. You probably have some relatives that have some of these. They're not all that rare. So do a little homework and things change. Again, science evolves. Not all physicians can keep up with it. Not any living human could keep up with all of anything anymore. Things move so quickly. So these markers exist, and these are markers for disease. There is a page in Wikipedia here. Let's have a peek on genetic disorders, and there are a bunch of them. A friend of mine from high school, Rothman, who got into human genetics, and he's a professor at Cornell. I haven't talked to Art in years, but he very useful for consulting. So we scroll down the page here in Wikipedia for genetic disorders, and lo and behold, we find a diagram really similar to the one that's in our textbook. And the thing with Wikipedia that's pretty cool is it's evergreen. That is, it's constantly updated. As new information comes in, people will go in and edit this and improve it. So most textbooks, I'm sorry to tell you, by the time they're in print, they're probably out of date. Sorry to say. And like many things now, short shelf life. So knowledge is evolving faster now than ever before. So not only does Wikipedia have more updated than a book possibly could, but they will also have pretty cool links to the literature. So if we zoom in here and look at sickle cell disease or phenylketonuria, you see this little superscript, that's a reference there. So if we tap on that reference, oh heavens, this one is 1986, so that's a little bit dated. Sometimes these aren't as up to date as they could be. So sometimes when a biochemist retires, they might go in and update some of these a little. Let's have a look at this one on cystic fibrosis, 2007, so that's a little more recent. Okay, so just know that those things exist and you can pursue that and look them up and find out if there's more up-to-date information for a particular thing, and there usually is. A lot of things are linked to the X chromosome, not as many to the Y chromosome. And the mitochondrial DNA. Thanks, Mom. So these all exist. That's the main point there. All right. So look up genetic markers as well in Wikipedia. They show some adorable little chicks here. And all kinds of different uses. Of course, they're used also in forensics to determine a crime scene or reconstruct history or something. And they break it into two classes, those which detect a variation at the gene product level, that is, genes produce eventually proteins. And then they also can find markers which detect variation at nucleotide changes that don't necessarily produce protein. So I did want to take a little peek in our book, in our chapter on genomics here, and notice there's mutations and polymorphisms. So poly, more than one form, so that can occur from genetics, from inheritance, but also mutations can and do occur all the time. When we talked about the electron transport system, we talked about free radicals or reactive oxygen species. Just as a consequence of breathing air, we get these free radicals and they attack DNA all the time, and we have enzymes that go along and repair that. And also ultraviolet light, UV light. You go out in the sun and you get some damage to your skin and your DNA. And there are chemicals and ionizing radiation. So look up, as I mentioned last time, what are some causes of mutations and what are they? So here's a little list of some types of mutations. Remember how when we use scantrons, I mentioned like frame shift mutation. So we talked about codons. Now we can sort of understand if you had a string of letters and you started reading in the wrong place, you might not get the triplets that code for the right amino acid. So that can happen from insertion and deletion. So frame shift is respect to reading the codons. It's the codon reading frame that we are talking about here. Okay, and they're also just point mutations. And sometimes, like I showed with one amino acid, we can change the third position and the wobble, and the amino acid doesn't change. The codon for the amino acid can be modified with a point mutation and it still produces the same amino acid. So here are two different codons where the third one has changed and it's gone from valine to valine, so no change. And then there's missense and nonsense, so they have different effects. All right, here's this really amazing lobster-like creature that has a 1 in 3 or 4 million chance of occurring. It's just this lovely color. And I am presuming that's while it's alive before it's cooked and displayed on a bed of noodles, with apologies to the vegetarians. So mutation is a change or an error in the DNA, and it can be passed along, carried along in replication. So it can be heritable. Heritable. Not all mutations are. Mutagen is a substance, or it can be radiation, especially ionizing radiation or heat or other things. So ionizing radiation, stuff like radioactivity. So there is a natural, there's cosmic rays going through us from birth to death. There's natural radioactivity. So if you eat bananas, you're getting some potassium-40 in there, and that's naturally radioactive, and it's banging away at your body. And then there's so-called man-made sources of radioactivity. So besides just substances, which would be chemicals, there can be all kinds of radiation. Of course, X-rays can cause mutations. And this is a big issue if one is exploring for breast cancer, because too many X-rays can actually produce breast cancer. So it's a double-edged sword. So you need to discuss these options with your healthcare provider. All right. Okay, a couple of good points about that. And next time we'll talk about recombinant DNA, or we'll talk about gene therapy. And we can also talk about GMOs, genetically modified organisms, which pretty interesting. So this, what they've shown here in the book with the sticky ends and all this, don't worry about all that detail. You're absolved from all of that detail. This is like so old school. Now, thanks to Jennifer Doudna, we've got CRISPR-Cas9. It's like a word processor. So let's just take a quick peek on Wikipedia here, or it can start in Google. I don't know how to, just in case I don't spell it right. So if I look in Wiki here and I get the spelling wrong, see, well, it did correct me, but Wiki doesn't, I added the E intentionally. Wiki doesn't always correct you as much as Google. So if you're uncertain, start with Google. But let's just dive into this, Wikipedia. So basically this goes back to the bacteriophages that I talked about. And it's essentially the two of these, CRISPR and Cas9, are gene editing tools, the way that a laptop or an iPad or something is a word editing tool. It used to be when you edited genes, we'd sort of like hack at them with axes, and we do little chunks of things at a time like this. And it was really painstaking and took a long time to do this. And it was very tedious. I would have gone into this area, but it was just too tedious. But now with CRISPR-Cas9, it's phenomenal. So look that up, and yeah, so see, CRISPR is an acronym, and the Cas9 protein for genetic modification. But I did just want to show you the picture of the T4 bacteriophage, just to show you how really amazing viruses are. So that's for the E. coli, Escherichia coli virus. And let's see, they've got to have a mugshot in here. Oh my heavens, unbelievable. They don't have a mugshot. So I'm going to get out of Wiki here and go to Google and put in the see, T4. I'll even do bacteriophage. Yeah, that's right ahead of us there. And we'll just click on images here. And these guys are freaking amazing. So here's an actual electron micrograph of one. And here's this diagram here, and it's pretty phenomenal. Icosahedral head, like, 20 side, 20 triangles. It's got these little landing gears that attach to the bacterial cell and this base plate, sheath, and collar. And it actually grabs hold of the cell, attaches, and drills and injects its nucleic acid head in there. It's really incredible. So look for YouTube on that. But just to show you that viruses, again, this is a bacteria, a virus that attacks bacteria. And it looks like a space invader. Mother Nature is very clever. So pretty amazing. All right, that about wraps it up for now. Look at the YouTubes I've posted and find some of your own. Try check one out on the C4 bacteriophage. Stay well, be healthy. Cheers. Bye-bye.